As we zoom in on missing DNA, we can see that it's made up of nitrogen bases. There are two types of nitrogenous bases: purines, which are adenine and guanine, and pyrimidines, which are cytosine, thymine, and uracil. Base pairing rules cause only adenine and thymine to pair and only guanine and cytosine to pair. Hydrogen bonds make sure of this because adenine and thymine are two hydrogen bonds and guanine and cytosine have three. Let's take a closer look at the phosphate backbone. The phosphate backbone in this diagram is encased in blue arrows. The yellow circles are the phosphoester bonds. At the 5' end, the phosphate group is connected to the DNA molecule, and at the 3' end, the DNA molecule is connected to the next phosphate group. DNA runs from the 5 to 3 prime, as the DNA can only be synthesized by adding to the 3' end. Therefore, each strand is anti-parallel, pointing in opposite directions. Let's take a closer look at the DNA molecule prime location. At 1', the carbon attaches to the base. At 2', we can determine whether the molecule is DNA or RNA. DNA molecules have no oxygen, as shown in the image, whereas RNA will have an oxygen at the 2' location. At 3 and 5' location, the phosphate group and the DNA molecule bond together. The phosphate groups have a negative charge, and the bases have no charge. This causes the phosphate group to be hydrophilic, and the bases to be hydrophobic. In order to avoid interacting with water, the bases will stack on top of each other. This is called base stacking. Here we can see the major and minor grooves. The major grooves have more space, which allows proteins to interact with the bases. DNA can be left or right-handed. You can determine this by using your hands. If the strand at the bottom of the DNA are slanting the same way as your left or right thumb, then the DNA is left or right-handed accordingly. The phosphate backbone of DNAs have a negative charge, which results in them pushing away from each other. However, when high salt concentration neutralizes the negative charge, there's less repulsion between the strands, making it harder for them to break apart. This causes the temperature of melting to rise, as it is now more difficult to break apart the strands. Here are my references.